أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم اقتربت الساعة وانشق القمر إن يروا آية يعرضوا ويقولوا السحر مستمر وكذبوا واتبعوا أهواءهم وكل أمر مستقر ولقد جاءهم من أنباء ما فيه مزدجر حكمة بالغة فما تغني النذر صدق الله العلي العظيم. Lecture number 13 for first year medical section. An important discussion once again. The discussion about the dynein arms. The dynein arms, a protein which moves the cilia, a specific protein, a special protein which is allocated just for the movement of the cilia. Cilia cannot move without that protein. Now how these dynein arms, how these 11 fibers are arranged, this is the today's discussion in this lecture. How they are arranged, because their arrangement is so specific. If their arrangement even minor percentage their arrangement will damage, the cilia is unable to move because the fixed position of dynein arms with the fibers will move the cilia and flagellum. If you will see, I just locate the movement of the cilia. I will just locate the location. Yes, this is the diagram you are watching, the dynein arms and the fibrils. You remember, do you remember the how many type of the fibrils were present? Do you remember in the last lecture? 11 fibrils were present in one cilium or in flagellum or in one flagellum. Now I will show you, these are 11 peripherals and these are peripherals. Peripheral mean at side area, and these are at the central. 2 fibrils at the center. The 9 fibrils peripheral mean they are at the sides, and that they are in the form of the pair. They are in the form of the pair. So these 9 doublet fibrils, one is inner fiber, one is outer fiber. They are attached with the dynein arms. Now question is, where the dynein arm is? That is a question. Yes, I just want to show you dynein arms. The dynein arms attached with the inner fiber, and this is another diagram showing peripheral fibers and inner fibers. I was mentioning spokes. Do you understand? In the last lecture I was telling that the central fiber attached with the peripheral fiber, just like the spokes of your spokes of the wheel of bicycle or motorbike. You see just spokes. These are the spokes. These are the spokes which are present. I can draw this diagram even. This is a fiber, peripheral fibers. These are 2 central fibers and they are attached by spokes. These are spokes. Okay ji. Now go back to another thing which is I think will be able to explain. This is a paramecium. Paramecium is a microorganism. You know, paramecium is a microorganism. Paramecium move by cilia. So we are taking one cilium. I was mentioning in the last lecture, this is not collective structure of all the cilia. I am just mentioning the one structure of the one cilium. So much special structure of one cilium. So if you will combine all these cilium structures, then you can imagine how much efforts, how much force these all cilia collectively can produce. If you will just see this cilium, you are taking one cilium. So one cilium you are taking one cilium, and this one cilium you will take, and this one cilium will go into this fibers, these fibers. 9, once again I am using the same terms again and again to memorize you students, the 9 double fibrils at the peripheral site and the two fibrils at the central site. Now I am just mentioning clearly how dynein arms helped in the movement of the cilia. I just show you the attachment of the dynein arms. You have to see this diagram very carefully because this will explain. Look here, red color protein is dynein arm, red color protein is the dynein arm. Similarly, if you will see, where is outer fiber, inner fiber, outer fiber, inner fiber? This is a outer fiber, this is a inner fiber. Between them dynein arms are present, and these dynein arms they produce energy. They produce energy, and energy comes by ATP. ATP give energy to dynein arms. Once again I will tell you, ATP hydrolyzed. What is hydrolysis? When anything react with the water, this is called hydrolysis. Where is ATP in the diagram? Where is ATP in the diagram? This is the ATP. ATP, you are watching. So ATP will hydrolyze, ATP will react with water, ATP will react with water, ADP will form, water will be released. No, water will not be released, because ATP react with water, ADP will be formed, and inorganic phosphate will be formed. Inorganic phosphate. Do you remember how ATP gives energy? Do you remember how many phosphates are present in ATP? That is another topic. In the next chapters we will study that ATP, how the ATP is able to produce the energy. ATP consists of 3 phosphates molecule. The last 2 phosphate bonds are very important because they store energy. If you break one phosphate bond, it changes to ATP. So ATP further can give energy by breaking another phosphate bond, because 2 energy rich bonds are present in the ATP. So what will happen? The ATP who will break the energized phosphate bonds, the dative bond, which is called energy bonds of the phosphate in the ATP molecule, who will break the water, will break water reaction with the ATP, hydrolysis. Reaction of the ATP with the water is called hydrolysis, HYDROLYSIS. So what will happen? The ATP will produce energy. This energy will be given to, it's very simple now. It will give energy to dynein arms. The dynein arms are attached with inner fibril, outer fibrils, and they will move the cilium. They will move the cilium. They will bend the cilium. They will exert the sliding force. They will exert the sliding force in the cilium and the flagella. You can see the sliding force. If you will compare these 2 diagrams, this is the figure number 1. This is the figure number 1, and this is another figure, figure number 2. So in these 2 figures, what difference you are observing? What difference you are observing, students? Because this is important. I just want your attention. One type of figure, the cilium, the fibrils are tilt towards the left side, or they are little bit bent towards left side. But first diagram, yani number 1 diagram, this diagram is showing the cilium are just straight. Question is, why the ciliums are straight here, and why the cilium bends? Are you watching the ATP molecules? These ATP molecules hydrolyzed, they react with water, they produced ATP and inorganic phosphate, energy was released, that energy was given to dynein arms. Dynein arms between the outer fiber, inner fibers, they moved, they put the force, they exert the sliding force. They exert the sliding force so that these fibers, which fibrils? Question is, which fibrils? Central fibrils or the peripheral fibrils? You know, in the previous lecture I was talking about the importance of peripheral fibrils. I was talking that the movement takes place by peripheral fibrils. The most importance I was giving the peripheral fibrils. These are peripheral fibrils. These are peripheral fibrils, and these are the peripheral fibers, and they will move the cilia and they will exert the force. They will cause effective stroke and recovery stroke. Now I just want to show the details which I just already told you, and the explanation. Dynein arms here, dynein arms written, and now adjacent double and exerts sliding force. These fibers which are arranged in the form of peripheral fibers, look here, this diagram will show you exactly arrangement. So if you will see this one, these are the 9 peripheral fibers. They are, how they are attached with each other? This is a question. How they are attached with each other? By dynein arms, by dynein arms. And simple 2 central fibrils. These are the spokes of the wheel. Spokes of the wheel explain the structure of cilium. So cilium consists of 9 peripheral fibers. Outer fiber is incomplete, inner fiber is complete, and between outer and inner fibers, dynein arm is present. And dynein arms actually one doublet, and other doublet are linked with each other by help of dynein arms, and they actually exert force. Red color were shown in diagram, dynein arms were labeled here. Actually they will exert the force by using ATP energy, and ATP energy will come from the glycogen, and then they will move the cilia, they will bend the cilia. The term I am using, when the cilia will bend, this is effective stroke. Important term I am using, students. When the cilia will bend, that is effective stroke. When the cilia will relax, that is the recovery stroke. So two stroke: effective stroke, recovery stroke, and effective stroke, recovery stroke. So there is some, now you will see the effective stroke and the recovery stroke, because I just wanted to show you. There are total 11 fibrils, total 11 fibrils. Now I will tell you what is effective stroke. 5 out of 9 double fibrils contract. Once again the question, who will contract? Who will contract the 5 fibrils? Who will contract the 5 fibrils? First five, they are in round form. First 5 fibrils, who will form? Dynein arm protein will move the first 5 fibrils. So when first 5 fibrils will contract due to force exerted by dynein arms, the first 5, the cilia will contract. Question is, sir, when first 5 fibrils will contract, whole cilium will contract? Cilia is one structure, it is just axoneme. So when cilia is bending, look here, this is very simple if I explain by my finger. Look here, when this cilium is this cilium, single cilium, my finger will bend, it means first fibers. The microscope tells us it consists of 11 fibrils. So central two are not involved in the movement because they are just giving the strength by spokes of the wheel attached with the peripheral fibrils. When the first 5 will contract due to dynein arms, it will bend, effective stroke. The last 4 will contract, it will relax. So all 9 fibrils will not work together. First 5 for contraction, the last 4 for the relaxation. I will continue this discussion in the next lecture. I hope you will understand, because I am revising the information again and again for your understanding. The paramecium is the best example to study effective and recovery stroke. First 5 contract, that is effective stroke. Last 4 contract. What will happen to last 4 when first 5 are contracting? First 5 contract, last 4 relax, effective stroke. Last 4 contract, first 5 will relax, recovery stroke. Thank you very much. Lecture number 13, we explain.